Zymogen activation. So, what are zymogens? Zymogens, or you can call them proenzymes, are inactive precursors of an enzyme. And for an enzyme to be active, it needs to undergo some biochemical changes, such as hydrolysis, to reveal the active site, or by changing the conformation or to remove the active, to reveal the active site. And it's only when this is done that the enzyme becomes active. So when we when we look at these zymogens, we begin to look at some specific things and how the enzymes shall operate. So to activate an enzyme, the active sites need to be reviewed. So in other words, we are saying that in zymogens, the active sites are not reviewed, right? So everything we do is to make this enzyme active, otherwise the enzyme will be inactive even though present catalysis will not take place. So zymogen activation is the process by which these proenzymes or these zymogens are converted from the inactive site to the active form. Okay, and this is done mostly by proteases. Okay, so the proteases are enzymes that cleaves proteins. So when they, in other words, we are saying that for zymogen to be activated, they need to be cleavages at some certain locations of the primary sequence of that enzyme. Now, for instance, you look at trypsinogen, you have an enteropeptidase that actually does some cleavages to form trypsin. Okay, so the inactive form of trypsin is the trypsinogen. Now, the trypsin can also help to convert trypsinogen to trypsin. And what is the function of this trypsin? Trypsin is one of the enzymes that we see in the digestion of proteins. And how do they do it? By helping to activate some other zymogens or some other proenzymes. For instance, it can be used in the conversion of chymotrypsinogen to chymotrypsin, proelastase to elastase, pro-carboxypeptidases to carboxypeptidases, and prolipases to lipases. So I have already mentioned them before. So when we look at insulin examples of zymogens, you have something like insulin. Okay, so the proinsulin is the inactive form of insulin. Fibrinogen is the inactive form of fibrin. Trypsinogen is the inactive form of trypsin. Chymotrypsin is the inactive form of chymotrypsinogen is the inactive form of chymotrypsin. Pepsinogen is the inactive form of pepsin. Proelastase, inactive form of prolactase. Prolipase are the inactive forms of lipase, and then procarboxypolipeptidase are the inactive form of carboxypolipeptidases. Okay, and when we look at insulin, okay, insulin, when it's formed from the messenger RNA, it contains 110 amino acids in the chain. But insulin on its own just has about 51, which means there are going to be some series of cleavages. So if you look at the picture on your right side, you begin to see the process. So when insulin is synthesized from the messenger RNA, it is synthesized in form of the pre-pro-insulin, and it has a signal sequence at the beginning of the N terminal of the amino acid. So of the polypeptide. So the first thing done is that the signal sequences are cleaved off. And when the signal sequences are cleaved up, there will be formation of disulfide bonds from the system molecules that you have in the sequence. Okay, so when you remove the signal sequence and the disulfide bondages are formed, the what you have is no longer pre-proinsulin, but this is the proinsulin which you can store in the body. But yet they are not active. So so this proinsulin cannot catalyze reactions. So what will happen is that there will be removal of the C chain. As you can see, you have the ABC chain. The B chain is in red, the A chain is in, I think that's yellow, and then the C chain is probably in ash form. So when the C chain is removed, you have only the chain B and chain A, which are now connected through disulfide linkages. That is the active form of the insulin, containing just about 51 amino acids. The rest have been cleaved off. So this is exactly what I was trying to explain. Okay. So the signal peptides from number one amino acid, which is methane, to the 24th amino acid where you have alanine, is being cleaved off. Then when you look at the proinsulin that is at the right, the you begin to see formation of disulfide linkages. These disulfide linkages are done by system molecules. Okay, about three of those linkages of the system that links itself. Now, after that, there will be the removal of the C chain. And then when you remove the C chain, you have your insulin. So and what I have just explained is what is written here. In insulin biosynthesis, how it begins with a messenger RNA to form preproinsulin, and then having a total of 110 amino acids. Okay, and then there will be a removal of the signal sequences. After that, there will be the formation of disulfide linkages, and after that, there will there will be a removal of the C chain, and what you're having remaining there is the B and the A chain only. Now, cryptocytes, chymotrypsin actually undergoes almost the same process. Okay, now there is the removal of there's a cleavage as position 13 and 16, thereby removing the amino acid 14 and 15. Okay, maybe look at it. So the truth is that whenever you're confused, you can go back to the previous, or you pause this video and then look at the places that are cleaved. Okay, the places that the amino acids that are cleaved are those ones in pink. So there's cleavage as position 15 and 13, so that amino acids at the position 14 and 15 are removed, which is serine and arginine. Okay, so there is also, after that, there is also a self-digestion at position 13, so that you remove these two. You have the threonine at position 147 and the asparagine at position 148. They will also be removed from the pi chymotrypsin. Now, when you remove this, you now have your alpha chymotrypsin, which is the active form. Now, why the pi chymotrypsin is also an active form of enzyme is that it helps in self digestion. Okay, now when you remove all these four amino acids, you can then have your active alpha chymotrypsin. Now, in summarizing whatever we have, we look at enzymes, ribozymes, and enzymes. Okay, not all enzymes are protein. So enzymes are antibodies that act like protein. They accelerate the rate of reaction. They, they get involved in the reaction, And then because the antibodies, we describe them as enzymes. We also have ribozymes, which are RNA molecules that help in speeding up rate of reaction. Now, enzymes can be synthesized, and when you synthesize an enzyme outside the living tissue, you can call such product synzymes. Okay, they are synthesized enzymes, so we call them synzymes. Okay, so